This is a guide of how you can perform the ELISA test for the Gliadin kit. It will give you all the information you need to do the test, including using Terra software. All TheraTest kit boxes are labeled with the content of the kit, the expiration date, and the kit's lot number. Allow all of the reagents being used to warm to room temperature, which is 18 to 25 degrees Celsius, for 30 minutes. Please note, the Gliadin assay should be run simultaneously with the TTG assay on separate plate frames. However, the reagents for each assay are not interchangeable. The reagents should be used separately for their respective assays. The kit contains a data sheet presenting the range of appropriate measurements that the calibrators and controls must fall within. The unit and ranges change with each new lot. Prepare all necessary equipment to perform the test. They are listed below. Upon opening a new kit box, label the caps of the calibrators, controls, and conjugates with the letter G for Gliadin in order to keep them separate from the TTG reagents. Prepare the 1X TTG/Gliadin wash buffer before beginning the assay. Using a graduated cylinder, combine 900 milliliters of deionized water and 100 milliliters of 10X TTG/Gliadin concentrated wash buffer. Following the preparation of the 1X TTG/Gliadin wash buffer, take the label from the 10X TTG/Gliadin wash buffer and place it on the newly prepared buffer. Initial and label the buffer with the date it was created. Label a dilution tube for the specimen diluent, positive controls, negative control, and each patient specimen. Place the labeled tubes into the dilution block in the location indicated on the plate map. Please note calibrators are pre-diluted, therefore the calibrators will not be diluted further. The 1X TTG/Gliadin wash buffer will serve as the specimen diluent for this assay. Add 1 milliliter of 1X TTG/Gliadin wash buffer to the dilution tube for the specimen diluent, positive controls, negative control, and all patient specimens. Using a new pipette tip for each sample, pipette 10 microliters of controls and patient specimens into their corresponding dilution tubes. Cap all samples firmly after adding specimens. Slowly invert the dilution tube rack 5 times in order to mix the samples. To ensure an adequate mix, watch as the fluid moves through the dilution tubes. Using a pair of scissors, carefully loosen and remove the caps from each sample tube. Label the dilution tube for the IgA and IgG calibrator. Place each dilution tube into the corresponding location within the dilution tube block, as presented below. Pipette 125 microliters of each calibrator into the labeled tubes. The calibrators are pre-diluted and should not be diluted further. On the left is an example of a plate prepared to run a Gliadin assay. On the right is a plate setup representing the sample layout for the Gliadin assay. Set a timer to 30 minutes. Using the multi-channel pipette, carefully drop 100 microliters of the IgA and IgG calibrators. Load the calibrators into their corresponding wells within the microplate. IgA calibrator into well B1 and IgG calibrator into well B2. Insert 7 pipette tips into the multi-channel pipette. Leave a space in the second channel of the multi-channel pipette as the calibrators have already been loaded into the microwell plate. Carefully drop 100 microliters of the specimen diluent, IgA positive control, negative control, and patient specimens. Load the specimen diluent, IgA positive control, negative control, and patient specimens to their corresponding wells in column A. Insert 7 new pipette tips into the multi-channel pipette. Exchange the IgA positive control with the IgG positive control in the dilution block. Carefully draw up 100 microliters of the specimen diluent, IgG positive control, negative control, and patient specimens. Load the samples into their corresponding wells in column B. Load any additional patient specimens into their corresponding wells. Start the timer that was set for 30 minutes. When the 30-minute timer sounds, carefully flick the microwell plate into the sink in a swift downward motion. Ensure the strips of the wells are firmly placed within the plate frame. This will serve as a precautionary measurement to prevent the wells from falling out of the frame and into the sink. Pour 1x TTG/Gliadin wash buffer into a clean reservoir. Set the multi-step pipette to number 5. This will load 250 microliters with each press of the lever. Insert 8 new pipette tips firmly. With the tips in the wash buffer, depress the multi-step pipette plunger to load the wash solution. Carefully pipette 250 microliters of 1x wash into each column. Lay the pipette down on the bench with the tips facing down and out. This will prevent any fluid from backing up into the pipette and contaminating it. Decant the wash into a sink. Repeat this wash procedure 2 additional times for each plate for a total of 3 washes. Firmly and carefully tap each plate upside down onto a paper towel. This is performed to remove as much excess wash as possible. Use new reagent reservoirs to load enzyme conjugates IgA and IgG. Pour the IgA and IgG conjugate into the reagent reservoirs. Use new pipette tips to load the conjugates. Set a timer for 30 minutes. Load 100 microliters of the IgA conjugate into the designated wells of the microplate. Load 100 microliters of IgG conjugate into the remaining wells of the microplate. This is an example of a microplate loaded with IgA and IgG conjugates. Any leftover conjugate may be poured back into the reagent bottle. Following the 30-minute incubation, wash the plate with the same technique as the first wash cycle. Wash each plate 3 times. Use a new reservoir for the addition of chromogen. A full plate of wells will require 10 milliliters of chromogen added to a reagent reservoir. For 2 plates of wells, add 20 milliliters of chromogen. Plan accordingly, as excess chromogen may not be poured back into the original bottle. Use new pipette tips to load the chromogen. Set a timer for 15 minutes. Start the timer as you begin to load chromogen into the first column of wells. Do not wait to start the timer until the entire plate is loaded with chromogen. Pipette 100 microliters of chromogen from a reagent reservoir into all of the wells. Try not to touch the edge of the wells with the pipette tips to avoid contamination. Never pour chromogen remaining in the reagent reservoir back into the reagent bottle. This will contaminate the unused chromogen in the reagent bottle. Simply discard any leftover chromogen. This is an example of wells containing chromogen following the designated 15-minute incubation period. Add stop reagent to the reservoir before the timer expires. Draw up 100 microliters of the stop reagent into new pipette tips. Pipette 100 microliters of stop reagent into all of the wells. Tap the side of the plates gently to mix the samples in the wells. The rest of the stop reagent may be poured back into the reagent bottle. This is an example of a completed Gliadin assay. The samples in each well are indicated. Place the developed plate into an ELISA reader for analysis. To read a new plate on Terra software, select File, Reading, New. Select Glia from the list of assays. Select Reader, Read and Collect. The reader will take 75 to 85 seconds to read the plate. This is an example of absorbance measurements displayed on Terrace software for the Gliadin assay. Cells A1 and A2 represent the diluent blank. Cells B1 and B2 represent calibrators IgA and IgG respectively. Cells C1 and C2 represent positive controls IgA and IgG respectively. Cells D1 and D2 represent the negative control. All cells that follow represent patient samples. After measuring the absorbance values, select the Sample IDs tab. Select Edit On. Enter the sample IDs from the assay in the same order the samples were loaded into the microplate wells. Select Save once all sample IDs have been entered. A new screen will pop up. Type in the name of the test that is run, followed by the date and initials of the individual who ran the test. Please see the example. Select OK to save the assay. This concludes the presentation provided by Theratest Laboratories in reference to performing the Gliadin ELISA assay, as well as a demonstration of test result storage within our proprietary software Terrace. If further clarification or assistance is required, feel free to contact our technical support department at 1-800-441-0771.